we're here at the Georgia Institute of Technology to just do a little bit more studying about this whole thing called Gel Electrophoresis. Now, what someone told me was that this technique was used to separate molecules of different sizes based on the pore size of the gel and based on the charge of the molecules, and it's just so freaking exciting. So, I think we're going to just sort of sneak into this lab and do a little, research of our own. Can't wait. We're here at the Georgia Institute of Technology in Georgia to discuss gel electrophoresis. Now, gel electrophoresis requires at least 4 tools. 1, we need a power supply. This power supply is going to allow you to create an electrical field for your protein. 2, we need a casting apparatus. Now, to create our gels, we don't simply just cut them out of a big mold of some sort. We actually have to cast them ourselves. And to do so, we use this tool.
Now, to cast a gel, you must first obtain agarose, measure out the specific amount that you need. Now, the pore size is going to be governed by how much agarose you actually add to your solution. Remember to clean up your area after you've used the balance. You then obtain the specific amount of running buffer that you need to make the specific percentage of gel that you want. Add your agarose to this gel and microwave it until it's dissolved. You then fix up your casting apparatus with comb, pour the agarose solution into the casting apparatus, set the comb, and you're done.
3, we need our protein, of course. Now, in this vial, we will find a bunch of different proteins, all with a negative charge. And lastly, we need our running tray. Now, our running tray will actually hook up to our power source. It has 2 electrodes, a negative electrode and a positive electrode. Once this is hooked up to our power supply, we will have an electrical field running through. So the Lorentz equation describes the force needed to move a charged molecule in a particular electric field. What does that mean? That means if we have a charged molecule, say, that's negative, we have an electric field which is governed by an anode, cathode, and an anode. We put our negatively charged molecule in this electrical field, then there's going to be a force created which pushes this molecule either this way or this way. Now, because this is a negatively charged protein, it's obviously going to move towards the cathode, because opposites attract.
Now, if all protein has the same charge, we're going to have to start to separate based on molecular size, which is governed by pore size. Okay, so now you know how charge influences the movement of a molecule. What about the pore size? Okay, well, here's an example. You see those trash cans at the end there? They represent a small pore size. Now, when the anode starts to push the negatively charged protein towards the trash can, guess what? It's too big, and it gets stuck. It can't go. But what about when it gets a bigger pore size? Yeah! It's able to get through just fine, very easily. So you see here, pore size influences whether or not small or large molecules are able to flow easily through the gel. It's quite simple. And of course, charge influences how fast it's going to move as well.
So let's get started. First thing we want to do is to remove the comb from our gel. Nice and slow, so you don't rip the gel. Once the comb is out, we remove the gel like so and place it in the running tray. Now we add our running buffer, which basically acts as a media to allow the charges to flow from one end to the other. It's now time to load our gel. We do that with one of our yellow pipettes. We take our protein. We add 20 microliters to one of the wells. Now this, you have to be very, very careful, very delicate. Go in to one of the wells. Put the tip right on top of the well and begin to release your protein slowly. Trying not to break the well. Keep the tip very still. We then at our top, since we know our protein is negatively charged, we put the negative electrode at the top so that it pushes the negatively charged protein away from the anode. Red goes into the positive charge, black goes into the negative charge, and we're ready to go. Alright, so there we go. Today we learned about the Lorentz equation, and we learned how to cast a gel, and we learned how to actually put this whole gel electrophoresis apparatus together. But what does it all mean? Well, you can figure that we can actually manipulate the pore size of our gel. We can manipulate the charge on our protein, and therefore we can actually separate a complex mixture of proteins any way we want. So we can purify, we can separate, and we can analyze. Gel electrophoresis is important. Trust me, it's important. But I think someone knows I'm here, so I better get going. See ya.